afternoon, students. After knowing the structure of DNA, we need to know how is this DNA transferred from one generation to the next generation. As DNA is the genetic code of life, it is very, very essential that the DNA which is transferred to the next generation is absolutely correct. The fidelity of this process, which is called as DNA replication, is mandatory as it is the blueprint of life. Any error in DNA is going to cause severe disease in the next generation. So today we are going to study about one of the very important topics of biology, that is DNA replication. In the last class, I told you about the central dogma, that how the DNA is transcribed into RNA and then RNA is translated into proteins. But what happens when you fall down or when you have grown from a small child to a young adult? The cells have divided, and so the DNA is also being transferred to the next cell. That means there is a continuous cell division and the DNA in the parent cell is transferred to the next generation. Now, let's understand some very basic important concept. For example, this is a parent cell. It has to transfer its DNA to the next two daughter cells after division. The amount of DNA which was present here was 23 pair of chromosomes. We get 23 chromosomes from our father and 23 chromosomes from our mother, and together they make 23 pair of chromosomes in each cell. Now, when this cell has to divide into two daughter cells, each daughter cell has to get 23 pair of chromosomes only. They just can't get 23 and 23, and subsequently if they divide, this number is going to reduce. The number of chromosomes is hallmark of any species, it cannot reduce. That means the amount of DNA which is present in the parent cell, it duplicates, it replicates, it doubles itself, and then the parent cell, it divides into two daughter cells retaining the same number of chromosomes. Now, the DNA which is present here has to be doubled, and then the cell is going to divide into two daughter cells through a phenomenon called as DNA replication. Now, I will be telling you what is DNA copying. Each cell division, cell must copy its entire DNA. So it's not so that before a cell division, the chromosome number 1, 4, 7, 8 is going to double itself and rest of the chromosomes stay like that. No. During DNA copying or DNA replication, all the chromosomes, all the DNA are going to duplicate. The amount of DNA in them is going to duplicate. So each daughter cells get a complete copy of genes. That means if there is 23 pair of chromosomes, each chromosome has to duplicate, and then the duplicated copies and the initial copy of this DNA is distributed equally into two daughter cells, confirming that they get a complete copy of genes. Now, the rate of synthesis in bacteria is 1000 bases per second. DNA replication is a fast process where the fidelity of this process is maintained. So it is a very efficient process where the speed, high rate of replication is maintained with approximately 99 or above fidelity. And in mammals, it's a bit slower because the errors cannot be afforded. It is around 100 bases per second. Now, the problem with single replication origin in DNA. Now, what is happening is that the DNA replication starts at one point in DNA, which is called as origin of replication. For example, this is a bacterial DNA which is circular. There is going to be a region of this DNA which is called as OD site, which is called as site where the DNA replication is going to initiate. So, there can be single replication origin or there could be multiple. Now bacterial genome, since it is small in size, is having just one or few origin of replication. However, the amount of DNA in humans is comparatively very high, and if it is going to work with one origin of replication, it is going to take lots of time. So there is multiple origin of replication in mammals. Now, DNA copying solutions, double helix has to be copied. That means, as I told you in the previous class, that both the strands of the DNA has to be copied. It's not so that only one strand has to be copied. And the DNA copying, during DNA copying, a directionality of DNA replication is followed. And since the direction of both the strands is opposite, one is from 5' to 3' and the other is from 3' to 5'. So, since DNA replication takes place in only one direction and the direction of both the strands are different, there has to be certain strategies to make sense of this process, how it is happening. Second is, semi-conservative solution exists. That means, it's not so that from both the strands of the DNA, for example, this is a parent strand of DNA. Now it starts replicating. This acts as a template for the new DNA, and this acts as a template for the new DNA. So each daughter cell is going to get one parent DNA and one newly synthesized DNA. Other daughter cell is going to get one parent DNA and one of its strands of its DNA is going to be newly synthesized DNA. It's not so that one of the newly formed DNA is composed of both the newly synthesized DNA and the other DNA strand or the duplex is consisting of both the parent strands. One strand is parent strand and the other strand is newly synthesized strand. That is why DNA replication is always semi-conservative. Means 50%, half of its DNA is parent DNA and half of its DNA is newly synthesized DNA. Unwind at one point and use that as origin of replication. I have told you that DNA is twisted around each other. For its information to be coded for the next generation, it has to be opened. And it starts at one point which is called as oricide, that is origin of replication. Now, as you can see that these are the two strands of the DNA, one from 5' to 3' and other from 3' to 5'. And this double helix, it is not like this, it is twisted. For ease it has been shown like this, and this is called as origin of replication. From here the DNA replication is going to start. So double helix is going to open here with the help of certain initial proteins which are going to bind at this point, like single strand binding proteins, helicases, etc. And from here the DNA replication is going to start. This particular site is called as origin of replication. Now you can see that this is an electron micrograph picture of DNA, where in original picture you can see there are certain open structures, O-like structures, and these are basically multiple origin of replication, so that the huge large amount of the DNA starts replicating from multiple points and the DNA replication can be completed in a possible manner in particular time. So this is origin of replication, and this is how it has been observed that with time the size of this O, it increases. Initially it was small and then it increases. It's showing that the DNA is replicating in both the direction, and it is not so that it is opening only from one direction, it is opening from both the direction. Now DNA copying solution. The region is at 80 rich to allow easy separation. As I told you that the origin of replication is going to be the first one where the DNA replication starts. So wherever the unwinding has to take place, it is mostly between in a particular region which is rich in 80 sequences. Why 80 sequences? Because in the last class I told you that A and T, they bond together with each other through double hydrogen bonds. So they can separate easily as compared to GC rich content because there are 3 hydrogen bonds in between them. So in eukaryotes, there is multiple origin of replication because the amount of DNA is very large. So the multiple origin of replication facilitates a quick replication of DNA in required time. Now, the parent strands in this particular structure is shown in yellow and the newly synthesized strands are shown in red. You can very well see the directionality of the parent strand, which is 5' to 3' in this strand and 3' to 5' in this strand. However, the newly synthesized DNA, which are synthesized, they are also anti-parallel to the parent strands. In front of the 5' there is going to be 3' and analogous to 3' there is going to be a 5'. Similar pattern can be observed here. So DNA replication follows one direction, which is always 5' to 3'. Since the direction of both the parent strands are separate, the way of replicating both the strands of the parent is going to be different in DNA replication. Now, this is a pictorial cartoon which is showing you that these are the bases and these are guanine and cytosine which are joined together by 3 hydrogen bonds. These are adenine and thymine which are joined together by 2 hydrogen bonds, and these are the 2 parent strands and which are in opposite direction. Now, when the DNA replication takes place, one of the parent strand is going to have the information of what all bases will be involved in the synthesis of the new daughter strand. Because if there is going to be A here in the parent strand, the daughter strands need to have T only, cannot have G or C. If there is G here, it has to C. It has to have C. If it is T here, this daughter strand needs to have A. So a rule of complementarity is followed. So whatever information is there in the parent strand, complementary information is transferred to the daughter strand. It is not so that haphazardly any bases are coming and they are forming the phosphodiester bond subsequently with these nucleotides and new daughter strand is synthesized. Incorporation of even one nucleotide is very precisely checked, because any error in the genetic material is going to be transferred from one generation to next generation. So fidelity of DNA replication is very, very high, above 99%. So for example, this is a parent strand and this is a newly synthesized strand which is getting replicated during the process of DNA replication. This is the nucleotide, this is the deoxyribose, this is the base, this is the 5' phosphate. The 3' hydroxyl of this nucleotide is going to form the phosphodiester bond with the 5' phosphate of this incoming nucleotide, which is having cytosine as its base. And this cytosine is forming the 3 hydrogen bonds with the guanine, which is a base of the nucleotide of the parent strand. And you can very easily see that even the newly synthesized daughter strand is anti-parallel. It is 3' and it is 5'. The another thing which is worth observing is that the complementarity is maintained. If it is G, the daughter strands will have C. If it is T, the daughter strand will have A. If it is G, it is going to have C. So it is not just incorporating any nucleotide of its choice. The rule of complementarity of the purines and pyrimidines is strictly followed, and that is how the incoming nucleotides keep on forming the phosphodiester bond with the previous nucleotide and is formed between the 3' hydroxyl of the deoxyribose of the previous nucleotide with the 5' phosphate of the incoming nucleotide. Now, what is DNA replication? DNA is copied by a replication machine that travels along each replication fork. Now, all of a sudden, why fork? When the DNA template opens at the origin of replication, since DNA replication takes place in 5' to 3' direction, there is a way these strands travel along the parent strand. The way they are replicated along the parent strand. And that particular structure has been named as replication fork. One of the most important members of this complex is DNA polymerase. The name itself says DNA polymerase, something which synthesizes DNA, polymerase, an enzyme which duplicates, which help in synthesis of DNA, and DNA polymerase is one of the most critical enzymes which is responsible for DNA replication. It is able to add DNA subunits and make the new DNA, that is the daughter DNA. So DNA polymerase, it was first discovered in 1956 by Sir Kornberg, and in bacteria E. coli. It is of three types: DNA pol 1, 2 and 3. DNA pol 3 is involved in DNA replication, whereas DNA pol 1, pol stands for polymerase, is responsible for repair of DNA. For example, there is a wrong base which is incorporated by DNA replication, then it has to be removed and right nucleotide has to be incorporated. So that is called as repair. That is checked by DNA polymerase 1. Humans have four types of DNA polymerases. 1, DNA alpha, DNA pol alpha, beta, gamma and epsilon. Alpha, beta and delta are the nuclear DNA and gamma is involved in the mitochondrial DNA. Now, that clearly shows that the enzymology for the DNA replication in prokaryotic system and eukaryotic system are different. But the principle which they follow for DNA replication is more or less the same. That means that a prokaryotic gene and eukaryotic genes are replicated through different enzymes. All the DNA pods have two properties. First, this is the basic principle of DNA replication, that the DNA replication always takes place in 5' to 3' direction. It can never take place in other direction. And second thing, only add to the end of the existing double-stranded DNA. Therefore, they cannot start synthesis of DNA from scratch. Means DNA polymerase need to have a free 3' hydroxyl group. All of a sudden the DNA separates and DNA polymerase comes and starts a replication. It cannot happen. It needs to get an overhang of 3' hydroxyl. It cannot start DNA replication by its own. That means some other enzyme is laying down a stretch of oligonucleotide before the action of DNA polymerase. And this is how it happens. These are the two strands. Yellow ones are the parent DNA strands, 5' to 3' and 3' to 5'. You can see that the DNA replication which is shown of the daughter strands in green strands is always taking place in 5' to 3' direction only, because DNA replication takes place in 5' to 3' direction only. This is the direction of fork. There were experiments which were done to show that this replication bubble is going to move in both the directions. It is not unidirectional, that one end is static and other one is dynamic. Both the ends start moving, and this is how the bubble it increases in size. Now, you can see that if this is 3', the daughter strand is going to be 5'. The DNA replication is going in this direction, that is 5' to 3'. If this is 3', the daughter strand will be 5'. So DNA replication will go in 5' to 3'. So the direction of DNA replication is exactly opposite, but the directionality is 5' to 3'. So that the DNA polymerase can add the nucleotides at the 3' end of the daughter strand. Now you can see this is a replication fork. Now, I've told you two important features of DNA polymerases. First is its directionality, that it is going to take place in 5' to 3' direction of the daughter strand. And second is that it is not going to start from scratch of any DNA. It needs to have a free 3' hydroxyl end. That means there need to be an enzyme which is laying down a stretch of oligonucleotide, and that enzyme is primase enzyme. Now here you can see the dark pink strand is of the parent strand, and they have opened up for the DNA replication to take place. Now since the two bases here, the complementary bases, are in a tendency of forming hydrogen bonds with each other, they need to come again together to form the spiral of double-stranded DNA. So they have to be kept apart, and they are kept apart with the help of two enzymes, that is helicases and SSB, that is single strand binding protein. The helicases is going to unbind the DNA, and SSB are going to stick to these individual strands to keep them sterically apart, so that the daughter parent strands coding is available for the synthesis of the daughter strands. Now then, the DNA polymerase cannot start working all of a sudden on these separated parent strands. You can see red strands overhangs there, stretch of red DNA. That is a stretch of 7 to 8 nucleotides, which is called as primer, laid down by the enzyme primase to provide a free 3' hydroxyl end to the DNA. After that red 3' hydroxyl end, the DNA polymerases are going to come and they are going to add on the fresh nucleotides, forming the subsequent phosphodiester bonds, as I have told you before. Now, same is happening here. The problem is that the DNA replication is going to take place from 5' direction to 3' direction of the daughter strand. There was the 5' strand, and now the DNA replication started. Now here what was happening, that initially the DNA was not that much open. For example, it was open till here only. The primer was laid down in 5' to 3' direction. Now here it was having a free 3' hydroxyl end. So DNA polymerase is going to add the nucleotides in this direction. Now when the DNA opens apart, there was no primer. Now DNA polymerase cannot add any nucleotide ahead, because here there is a free phosphate group and there is no 3' hydroxyl group. Again primase enzyme will come and lay down a primer, that is a stretch of 7 to 8 nucleotides, so that it has free 3' hydroxyl end, and the new nucleotides will be incorporated by DNA polymerase. Same thing will continue to happen. So one strand of during DNA replication, one strand is going to be synthesized continuously, because as and when this is going, the parent strands are going to unwind, the 3' hydroxyl end is free and the DNA polymerase will add on the nucleotides. However, in the other strand, the story is completely different. As and when it opens, there is no free hydroxyl group. So every time the primase enzyme is going to come, it is going to lay down a primer which is going to have free 3' hydroxyl end, and the nucleotides are going to be added on. Now, these primers are basically made up of RNA. We really cannot afford these RNA parts in DNA. It has to be removed. So these parts are removed by another DNA polymerase, and the new DNA nucleotides are added, and then the phosphodiester bond is formed between both of them by the enzyme DNA ligase. So this process continues. I have told you that the DNA is spiral. If this is the bacterial DNA and I keep on opening it from one end, there is going to be a point when these two are going to be completely interlocked. So two strands have to be separated, and this is going to increase the stress. So what will happen? That one of the DNA strand has to be nicked, it has to be unwinded, and it has to be ligated back to the parent strand. This is done with the help of the enzyme topoisomerases. And the two daughter strands which are synthesized after DNA replication, they are separated finally with the help of these topoisomerases, which is type 2. So these are called as catenanes, and then they are separated. So steric hindrance which is produced because of the overwinding of the DNA, because of their extreme opening, is that stress is resolved with the help of enzyme topoisomerases. In this whole process, wherever the wrong base is going to be incorporated, that is removed by the phenomenon of link translation with the help of DNA polymerases. So fidelity of DNA replication is maintained throughout the process, and a series of enzymes work one after the another for the complete DNA to be replicated. So the flow chart goes as follows, that DNA replication is going to start at the origin of replication. Then DNA helicases separate the two strands, forming the replication fork. The DNA is twined around each other. It has to be separated. So that separation starts taking place with the help of the enzyme DNA helicase. Now, the DNA polymerase cannot start DNA replication of its own. It needs 3' hydroxyl group. For that, an oligonucleotide is laid down by the enzyme primase, and that is called as primer. The primer binds at the replication fork, and the primase synthesizes the RNA primers. Then the DNA polymerases are going to come, and they are going to synthesize both the daughter strands in 5' to 3' direction, one in leading strand and other in lagging strand. Now, you can see that these fragments which are formed in the lagging strands are in parts, and these fragments are called as Okazaki fragments, are named after the name of the discoverer. So in one strand, that is in the leading strand, DNA replication is continuous, while in the other strand it is discontinuous, and it is in the form of the Okazaki fragments, and they are therefore named as the leading strand and lagging strand. Now, the DNA polymerase uses a primer to synthesize both the two strands, and when using the leading strand as a template, DNA polymerases add nucleotides to the new strand continuously. That is why it is called as leading strand, because every time the DNA reopens, the free 3' hydroxylane is always available, and DNA polymerase continues to add the new nucleotides. However, in the other strands, as and when the DNA reopens, there is no free 3' hydroxylane, and every time the primer has to be laid down by the primase enzyme, and thereafter the DNA is replicated in parts, in fragments, and these fragments are called as Okazaki fragments. The RNA primers finally cannot be afforded in DNA, and they have to be removed, and these RNA primers are removed, and the sequences are then synthesized by another DNA polymerases, and then the two new DNAs are the nucleotides of the two parts of the DNA are sealed by formation of a phosphodiester bond by DNA ligase enzyme, and that is how DNA replication takes place in prokaryotic and eukaryotic systems.